Hey guys, my name is Shreyas and welcome to Simple Biology. Today we're going to be talking about lipids. Now, a brief overview on lipids. Lipids are not true polymers because they aren't made up of monomers. In order for for a molecule to be considered a polymer, it must be made up of monomers, but lipids don't have monomers. So they're not true polymers, but they're still considered a type of biomolecule. Now, there's many types of lipids. We're going to be actually talking about, I think, 4 different types of lipids, and they're all very different from each other, but they're all grouped together because they are all hydrophobic. Now, if you don't know what this word means, I highly recommend that you go back and watch our whole unit on chemistry that we have attached to this course. It's the first unit, and there's a video called the hydrophobic interaction where we talk about this, but just as a reminder in case you forgot, hydrophobic means water-fearing. So a molecule which is hydrophobic will not mix well with water. So all lipids are grouped together because they are all hydrophobic. Now, the first type of lipid I want to talk about are fats. Fats are constructed from glycerol and fatty acids. Let's first talk about what glycerol is. Glycerol is this compound that I have drawn up here. It has 3 carbon atoms, and it has a hydrogen atoms, and then it has 3 hydroxyl groups here. That's glycerol, this red molecule right here. Now, a fatty acid is a large hydrocarbon attached to a carboxyl group. I've drawn an example of a fatty acid right here. If you notice, we have a very long hydrocarbon here. If you look, we have this carbon, and then a very long unbranched hydrocarbon. You should know what that means, unbranched. If you don't, you need to go back and review previous videos. So we have a very long unbranched hydrocarbon, and at the very end of that, we have a carboxyl group. Now remember from the video on functional groups, whenever a molecule has a carboxyl group, it makes that molecule an acid. So that's why this molecule here is called a fatty acid. You can think of the fatty as the large hydrocarbon because it's fat, as in it has like a lot of atoms, and it's an acid because it has this carboxyl group at the end. So what happens in order for a fat to be created, a bond between a fatty acid and a glycerol is created, and that's called a ester linkage, and it's created by dehydration synthesis. Now let me go ahead and explain to you how that happens. We've talked about dehydration synthesis. Now let's actually see it in action. So if you look here, what's going to happen again, a fat is a glycerol and fatty acids. So they're going to have to react with each other in order to create a fat. So what's going to happen is that the hydrogen atom right here is going to react with the hydroxyl group right here. What will happen then is these atoms will all react, and then they will create water. And that's why it's a dehydration synthesis, because water is being expelled. Let me go ahead and draw that. So water is being expelled. And then what will happen is a bond will be created like this. So now that the water molecule is gone, there'll just be one single bond like this. And now that this fatty acid is bonded to the glycerol, we have this whole molecule right here now, because these two parts are bonded together. This whole molecule is now a fat. And this linkage right here is called an ester linkage. Now, triglycerides are a very common type of fat, and they're fats with 3 fatty acids. Here, this fat right here, which we have created, has only 1 fatty acid. But what can happen is another fatty acid can react with this hydroxyl group right here in the same fashion, and another fatty acid can react with this hydroxyl group here in the same fashion. So whenever a fat has 3 fatty acids, not just 1, it's called either triacylglycerol or triglyceride. Okay, so that's the first type of lipid. Now let's go ahead and let's discuss this first before we discuss the second type of lipid. The difference between a saturated and a unsaturated fat. This is very important in biology. A saturated fat is a fat with no double bonds. We have a saturated fat right here. I've never shown you the line structure before, but if you look at this, this is a very simple way of drawing a hydrocarbon chain that scientists use all the time. Because drawing something like this with many carbon and hydrogen atoms is very time consuming. This actually took me almost 5 minutes to draw. So what scientists do instead is they just draw this line. So at every single, if you look at this jagged line right here, at every single point here, there's a point, there's a point there, there's a point there, there's a point here, there's a point here, there's a point here, there's a point here. At every single point is a carbon atom attached to hydrogen atoms. So basically this line right here is just showing the same thing like this, a very long hydrocarbon chain. So, and if you ever see a line like this is a very long hydrocarbon chain. That's all it is. Okay, and this right here is a triglycerol, or a triglyceride, because it has 3 fatty acids. It has one here, one here, one here, so 3. Okay, so remember this. If you ever see this in a biology textbook, a jagged line, and you'll see this in organic chemistry and down the road. And I'm going to be starting to use this because I don't want to take the time to draw this, something like this. All this means is that we have a hydrocarbon chain. All right. So a saturated fat is a fat with no double bonds, and if you look here, there's no double bonds, and that's going to make more sense when I talk about an unsaturated fat. An unsaturated fat is fat with one or more double bonds. Okay, so if you look here, we have two hydrocarbon chains which have no double bonds. But what happens here is that we have a double bond. If you look here, there's 2 lines. So this represents a double bond. Again, I didn't want to take the time to do that, to actually draw every single carbon atom. But what happens is that when we have a double bond like this between carbon atoms, the hydrocarbon chain is no longer straight. If you look here, after the double bond, the hydrocarbon chain tends to kink like this. It's not straight anymore. Okay, and that's unlike the saturated fat. If you look at the saturated fat, it's just a very long parallel jagged line. But with whenever there's a double bond, you're going to start seeing these kinks where the hydrocarbon chain starts to bend like this. Now, the reason this is important is in saturated fats, because there are no double bonds in all these these fatty acids are straight, they can pack together very tightly. If you have one fat and you have another fat on top of that and you have another fat on top of it, because these fatty acid chain tails, or these hydrocarbon, you can either call it a fatty acid chain tail or the hydrocarbon tail, whatever you want to call it. It's the same thing. But because they're straight, they're able to pack on top of each other, and pack each other tightly, on top of each other tightly. So because they're able to pack on top of each other tightly, they're a solid at room temperature. Now, unsaturated fats, because they have these kinks, it's harder for these fats to pack on top of each other neatly, because of this jagged structure right here. So since they're not able to pack on top of each other tightly, they're liquid at room temperature because it's like less dense. All right. Now the reason why this is important to biology is because typically animal fats are saturated fats, and they're solid, but plants and fish fats are unsaturated. Okay. And it makes sense that animals have. For plants, it doesn't really matter. If plants want to store, I need to mention this. Fat is a very big reserve of energy for any organism. So for plants, since plants are stationary, it doesn't really matter if the fat is in a liquid form or a solid form. So that's why plants have unsaturated fats, because for a plant, since a plant is staying in one place its whole life, it doesn't matter if it's liquid or solid. But for animals, since animals have to move a lot, it makes more sense for animal fats to be saturated, because if it's saturated, the fats can be more densely packed and it's less weight for the animal to move. Okay, so that's why animal fats are always saturated, because it allows for the animal to be to store energy in less space. While that doesn't matter for a plant because a plant is stationary, it does matter for an animal. So that's why animals have saturated fats and plants have unsaturated fats. Now, there's two in animals. This will probably be mentioned later when we do our unit on animals and humans, but it's good to know, I guess. In animals, this saturated fat, this highly solid packed fat, is found in things called adipose cells. Just a fact that we will repeat later down the road. Sorry, but I guess it's good for you to know. In an animal, fat is found in adipose cells, and I already mentioned that fats are a great source of energy for organisms, but it also plays an important role in insulating organisms. For example, if you look at a whale will have a very dense pack of adipose cells, which will keep it warm even when it's in cold water. You've probably heard of that. It's typically referred to as blubber or terms like that, but blubber is essentially just fat stored in adipose cells, which helps it insulate and not get hypothermia. All right. Now let's talk about the next type of lipids, sorry, and these are phospholipids. Phospholipids are made up of hydrophobic tails and a hydrophilic head. So I have a phospholipid right here. So if you look here again, we have our fatty acid chain tails like this. But then we also have this huge head. Now I draw this out of proportion because I want you to be able to actually look at this molecule. Typically, the hydrophobic tail is going to be much bigger than the head, but I've zoomed in the head so we can look at the actual molecules in here. Now, the fatty acid part of the phospholipid is going to be hydrophobic because it is basically nonpolar, so it's not going to mix well with water. These tails right here are going to be hydrophobic, but the head right here up here is going to be hydrophilic, meaning it's going to mix well with water. Because if you look here, we have nitrogen here bonded to 3 methyl groups, giving it a partial positive charge. So since the nitrogen has a partial positive charge, it's going to be susceptible to mixing well with water. And we also have a phosphate group with an oxygen which has a partial negative charge. So that partial negative charge is also going to make it susceptible to mixing well with water. Okay, so now that this phospholipid displays this interesting property because it has one portion which is hydrophilic, the hydrophilic head, and then it has another portion which is hydrophobic, the hydrophobic tails. And because of that, phospholipids play a very important role in cell membranes. And I'm going to briefly discuss that, but when we talk about cells, this will be mentioned again. So if you look here, every single circle represents a phospholipid, and then these red lines are the circle is the hydrophilic head, and then these red lines are the hydrophobic tails. So what happens is these phospholipids will congregate like this. So all the hydrophilic heads will come on the outside like this and interact well with the water, and then all the hydrophobic tails will all congregate together and come on the inside. So what happens is now we have a very interesting membrane, because we have water on this side, we have water on this side, but we're able to separate that with all these phospholipids. These are hydrophilic, so they will be, these heads are hydrophilic, so they will mix well with the water and they'll be on the outside, and the hydrophobic tails don't want to mix well with the water, so they'll be on the inside. And then we have, eventually, I have just a portion of this, but if you look at a cell, we'll eventually have this nice membrane, because if this whole structure will form a huge circle like this, and then we have water on the outside, we have water on the inside, but then we have this nice membrane. And we'll discuss that later in biology when you talk about cells, but this is important to know. The last type of lipids are steroids, and steroids are lipids which have a carbon skeleton with 4 fused carbon rings. We talked about rings briefly in our video on hydrocarbons, but again we're going to see these rings again. And just like we have these jagged lines to represent hydrocarbon tails, in this we also have these rings in biology and in organic chemistry to represent carbon rings. We have these hexagons and shapes like that. So lipids are a carbon skeleton which have 4 fused carbon rings. So if you look here, we have one carbon ring right here, this hexagon, and then we have another carbon ring right here, and we have another carbon ring right here, and then we have another carbon ring right here. So they're all fused together. So a steroid has 4 fused carbon rings. And many hormones are steroids, and we're going to be talking about hormones way down the road, but know this fact. And cholesterol is an important steroid. Right here, this molecule right here is actually cholesterol, and it's found in animal cell membranes, and we're going to be talking about that in cells, but important to know for now. And other steroids are synthesized from cholesterol. So you've probably heard of steroids being substances that athletes use in order to gain muscle really fast, but most steroids are synthesized from cholesterol. You can think of cholesterol as the precursor for other steroids, and that's another important fact you need to know about steroids. But that's it for now. Lipids are as simple as that.